Chapter 1. What does aging really do inside our cells? Picture this clearly for a moment. You're looking into a mirror one morning and notice soft lines around your ioslim threads that weren't always there. Gradually, those faint lines deepen and multiply, carving into your skin like rivers slowly shaping land. While we see these outward changes clearly, beneath your skin, an invisible process hums louder each year. Countless tiny clocks ticking silently within every single cell of your body, marking the slow journey through biological time. Aging isn't a sudden switch that flips when you reach a particular birthday. Instead, it's the slow accumulation of countless microscopic errors gradually adding up. Like rust quietly stealing strength from steel or drops of water, patiently eroding stone, these tiny mistakes slowly wear our cells down from the inside out. Take telomeres, for example. At the ends of our chromosomes, those tightly coiled structures that store our genetic instruction set protective caps called telomeres. You can imagine them like the plastic tips on your shoelaces, keeping shoelaces from fraying. With each cell division, your chromosomes copy themselves, and these protective tips shorten a tiny bit. After many divisions, these telomeres become dangerously short, fraying away until the cell can no longer properly copy its DNA. At a certain point, when the cell senses its telomeres are critically short, it stops dividing altogether. Scientists call these cells senescent cells, but a more vivid picture may be zombie cells. Senescence is a strange, zombie-like state in which cells remain alive but refuse to grow, divide, or function normally. They sit stubbornly in place, sending out inflammatory signals to their neighbors, disrupting nearby healthy cells neither truly alive nor neatly dead. The accumulation of these zombie cells contributes significantly to the physical decline we associate with aging. Imagine filling a town, gradually, with more residents who refuse to work or cooperate, creating chaos, confusion, and waste. That's precisely what these senescent cells do in your body, spreading chronic inflammation, interfering with healthy tissue, and eroding biological functions. Meanwhile, within each of your cells, another subtle form of damage quietly accumulates: misfolded proteins. Proteins, the vital working machines inside our cells, have very specific shapes they must maintain. For proteins, shape is critical. Like carefully designed puzzle pieces, they work perfectly when folded correctly. But over time, damaged proteins misfold, they lose their proper shape and functionality, clumping together into sticky masses. Picture machinery inside a watch gummed up by debris. Misfolded proteins form these clumps inside your tissues, from your brain and heart to your muscles and bones, slowing or stopping essential biological processes. Diseases like Alzheimer's, for instance, arise partly due to the buildup of these sticky protein aggregates. It's a quiet, internal mess hidden completely beneath your skin. DNA itself isn't immune either. Your cell's genetic blueprint accumulates tiny mutations day by day. DNA damage comes from a variety of sources: ultraviolet rays from sunlight, smoke from cigarettes, environmental toxins, radiation, and even just normal, everyday cellular metabolism. Your cells constantly repair their DNA, but over years and decades, the repair machinery itself can become less precise, allowing mistakes to pile up. It's as if the original, carefully checked document of your genetic information slowly accumulates more and more printing errors, turning into a flawed manuscript over time. Each error might seem minor, but together they raise the risk of diseases and weaken the cell's ability to function. To make this clearer, let's step back to something relatable. Imagine your smartphone as it ages. When you first buy your phone, it runs fast. Apps open instantly. Everything responds quickly. But as time passes, hidden processes start running in the background unnoticed. Apps continue accumulating tiny memory leaks. Unnecessary files fill your storage. You never see exactly what's wrong, but eventually your phone slows down dramatically. No matter how careful you are, The gradual slowdown didn't happen overnight. It's a result of countless small inefficiencies quietly accumulating in the background over months or years. Aging works in similar patterns within our cells. It's not about a single obvious factor. It's many small slowdowns coming from damaged proteins, short telomeres, senescent cells, and DNA errors. Because of this complexity, scientists often talk about two types of age: chronological age and biological age. Chronological age, the kind we celebrate during birthdays, measures simply how many times the Earth has orbited the Sun since your birth. Biological age is trickier. It's the cellular age, determined by measurable molecular markers. You might have heard about the concept of biological clocks. One particularly intriguing method is the epigenetic clock. This fascinating molecular clock doesn't count years. Instead, it looks at tiny chemical tags on your DNA itself. These tags, called methylation marks, help control which genes are active or silent, and their patterns reliably predict an individual's internal biological age. Intriguingly, even two people sharing identical chronological age can have different biological ages. One body could be biologically older or younger based on genetics, lifestyle, and environmental exposures. This reveals another layer in understanding aging, helping explain why some people seem to age more gracefully than others, even when born in the same year. To envision what deeply aged cells might look like, consider Jean Calment, the oldest verified human ever to live, a French woman who reached a remarkable age of 122. Jean seemed cheerful and sharp into remarkably advanced years, but think for a moment about the microscopic reality inside her cells. While outwardly, Jeannie smiled and enjoyed her chocolate and wine, inside her tissues, her telomeres had become extremely short, her cells filled with misfolded proteins, her DNA riddled with tiny accumulated mistakes. Yet somehow, her system had maintained a tricky balance just long enough, postponing the onset of serious diseases. The exact factors allowing Jean Calment and others reaching unusual longevity to keep damage at bay still puzzle scientists today. Understanding exactly what aging does inside our cells is more than scientific curiosity. It's crucial groundwork if we ever hope to counteract, slow, or reverse the aging process. Exciting research has already shown that not all cells experience irreversible damage. Some deterioration, interestingly enough, is fixable or at least manageable. For instance, scientists have successfully removed senescent zombie cells from aging mice, dramatically improving their health and lifespan. It raises a hopeful thought. Perhaps the process we imagine as inevitable might someday become something we actively manage or control. We might even find clues looking beyond humans to other species that seem to brush off or resist aging altogether. Some animals appear uniquely adept at avoiding certain biological pitfalls we face. Creatures like certain turtles and naked mole rats hardly show significant signs of biological decline. How do they do it? Could they be holding secrets within their cellular biology to inspire innovative strategies against human aging? In coming chapters, we'll turn our curiosity closer to these amazing animals, unpacking precisely how they dodge some of the troubles that plague aging human bodies and what we might learn from their cells. This brings us to a compelling question. If turtles and mole rats can delay or nearly avoid these destructive cellular processes, can we? Chapter 2. Meet the animals that barely age. In 2007, off the icy northern coast of Alaska, a team of scientists studying bowhead whales encountered something extraordinary. Among these massive, gentle creatures swimming calmly beneath the frozen surface, they spotted a whale with scars from harpoonsa, unique style of harpoon last used by whalers back in the late 1800s. Curious about its true age, scientists carefully examined layers within its eye lens, much like the rings in tree trunks. What they discovered was stunning. The bowhead whale was over 130 years old. In fact, some bowheads swimming today have lived as long as 200 years, outlasting human inventions such as the light bulbs illuminating Times Square. Imagine a single animal quietly drifting through centuries, observing the human world change above, from sailboats to cruise ships, wood fires to neon lights, yet remaining steadfast and unchanged beneath the waves. This whale invites us to reconsider what aging truly is and how it affects different lives. But the bowhead whale isn't alone in its astonishing longevity. Traveling eastward across the frigid waters north of Iceland and Greenland, we encounter an even stranger creature, the Greenland shark. This large, slow-moving predator dwells in the freezing depths and is barely noticeable as it swims at a lethargic pace. Yet, hidden beneath its sluggish movements, the Greenland shark holds a monumental secret. Its amazing lifespan can stretch beyond 400, possibly even 500 years. Think about this. When the shark currently swimming deep below the Arctic surface was born, Galileo was observing planets, and Shakespeare was producing plays. Across centuries of cold darkness, these sharks have survived wars, explorers, and changing climates, aging at a glacial pace far slower than human biology. Scientists refer to species like the bowhead whale and Greenland shark, whose likelihood of death hardly increases over the years, as exhibiting negligible senescence. Negligible senescence is a fancy term, meaning that the animal's health doesn't steadily deteriorate with passing time. Its death simply isn't governed much by aging at all. Instead, outside factors like disease, predation, or starvation usually determine when these creatures eventually die, not intrinsic old age. For most animals, including humans, growing older is inevitably linked to weakening cells, less robust tissues, and declining vitality. But creatures demonstrating negligible senescence show us aging isn't biologically inevitable. Take, for example, the tiny freshwater hydra, a delicate polyp barely longer than your fingernail, with a transparent body and waving tentacles drifting under pond surfaces. While the hydra may seem modest and fragile, inside it lives one of nature's most powerful anti-aging secrets: hyperactive stem cells. Hydra continuously refresh their tissues by constantly dividing and replacing their cells, maintaining an endless state of renewal. If you were to cut a hydra in half, it wouldn't die instead. You'd end up with two functioning, complete hydras. This regeneration capability means hydra do not appear to age or deteriorate. Under laboratory conditions, a hydra can keep dividing and regenerating essentially forever. In reality, it may have few natural limits to its life. What scientists want to know is exactly how hydras keep these cells indefinitely active without malfunction or turning cancerous. Unlocking that puzzle could profoundly affect our understanding of human aging too. One of the factors uniting these remarkably long-lived animals, whether massive whales swimming frigid ocean currents or the tiny simple hydra floating quietly in freshwater, is their slow metabolic pace. Metabolism is our body's process of converting food into energy. The higher an animal's metabolism, the faster its tissues experience stress, damage, and wear. Lower metabolism is like an engine idling rather than racing full throttle. It burns energy slowly and smoothly, producing less waste and fewer damaging reactions inside cells. This metabolic slowdown is partly why Greenland sharks and bowhead whales, plunged into near-freezing waters, can survive centuries of gradual, gentle aging. Lower temperatures help reduce the molecular chaos and oxidative damage inside their bodies, providing extended, healthy lifespans. It's as if their cells remain quiet, stable, simply existing without rushing anxiously through the passage of time. Yet, remarkably long lifespan isn't simply about slow living or chilled temperatures. Consider the naked mole rat, a curious little rodent living deep underground in colonies beneath African deserts. Despite its humble appearance and small size, the length of your hand, the naked mole rat can live upwards of 30 years, which is extraordinarily long for a rodent. These small, wrinkly creatures rarely develop the chronic illnesses we associate with aging, especially cancer. What's their secret? Naked mole rats possess special oversensitive alarms in their cells. If any cell even slightly begins showing suspicious signs of replicating uncontrollably, capable of developing into cancer, those hyper-alert molecular detectors launch a self-destruction process, known scientifically as apoptosis, immediately removing the threat. This rapid, preemptive elimination of damaged cells means cancer never gets the chance to take hold. Another fascinating longevity champion lies submerged along chilly ocean floors, the rockfish. These colorful underwater dwellers may appear quite ordinary at first glance, yet certain rockfish species can live extraordinarily long, up to 200 years. What makes their lifespan possible is their brilliant genetic skill in keeping proteins folded correctly. Proteins are critical molecules that carry out nearly every function inside cells, but when proteins become misfolded, imagine paper origami that unravels. They become useless or damaging. Over decades, accumulated misfolded proteins cause cellular stress, disease, and aging. But rockfish genes produce special molecular guides, called chaperone proteins, to constantly ensure perfect protein folding and prevent harmful buildup. In this careful incremental management lies the secret to living 2 centuries beneath ocean waters without rapid decline. Long-lived animals share another distinctive biological trait. They repair their DNA extremely efficiently. DNA constantly suffers damage every day. Ultraviolet radiation, pollutants, cell division, ordinary body heat, and oxygen reactions all chip away at the integrity of the genetic code. Humans repair DNA too, of course, but inevitably, errors still accumulate gradually, leading to aging and disease. Animals with negligible senescence, however, possess far more robust DNA damage repair processes. They swiftly identify mistakes, fix them accurately, and maintain genetic integrity across prolonged lifespans. Even highly active animals, those with rapid metabolism and intense lifestyles, possess longevity methods we can learn from. The common bat, thrusting itself nightly through air at tremendous speeds, using vast amounts of energy, actually presents another fascinating example. Despite their frenetic pace, bats live exceptionally long. Considering their tiny size, some species surpass 3 decades. Their secret? Bats have evolved powerful antioxidant systems within their cells that neutralize reactive, damaging molecules called free radicals. These highly reactive particles normally tear apart cellular structures, yet bats efficiently mop them up, erasing the damage caused by their high-energy, oxygen-intensive lifestyles. Immune system differences also stand out in many of these durable animals. Long-lived species regularly display unique immune responses that effectively combat potential threats without causing chronic inflammation. Chronic inflammation, persistent, quiet irritation of the tissues, a major contributor to aging in humans. By contrast, longevity champions like bats, naked mole rats, bowhead whales, and Greenland sharks avoid unnecessary inflammation by keeping their immune reactions precise and surgical, maintaining calm within their tissues. Remarkably, we find parallels to these animal strategies within certain human populations as well. In Okinawa, Japan, where an unusually high number of people live beyond 100, scientists have noted biological similarities with long-lived animals like naked mole rats. Okinawans often have significantly lower insulin-like growth factor signaling, a molecular pathway that, when reduced, tends to prolong cellular health. Like the naked mole rats, Okinawans naturally keep insulin signaling minimal through diet and lifestyle, maintaining cells healthier and less susceptible to aging-related diseases. As we navigate the secrets uncovered by nature's oldest animals, one lesson emerges with clarity. Nature has already established effective biological instructions for healthy aging. Negligible senescence and extraordinary lifespans found naturally in diverse species, from gentle giants swimming for centuries under polar ice, tiny polyps endlessly regenerating in ponds, and rodents cleverly avoiding cancer tell us aging gracefully is biologically achievable and perhaps modifiable. And importantly, they debunk the common myth that size determines lifespan. Take the tiny ocean quahog clam with its 500 years of quiet underwater life, hardly bigger than your palm. Clearly, immense body size isn't required for great age, only finely tuned biology and cellular defenses. Through understanding these animals, scientists are discovering how lifestyle choices like reduced calorie intake, antioxidants, controlled insulin responses, and precise immune responses might mimic natural longevity strategies for humans. These remarkable organisms offer insight that may guide us toward healthier, longer human lifespans. But how do we bring these lessons into practical choices of our daily lives? Up next, we'll explore real, actionable habits and foods you can begin adopting today to enhance your own biological pathways, borrowing nature's ingenious tricks to foster lasting vitality. Stay with us, because your own longevity toolkit awaits. Chapter 3. Lifestyle Hacks That Slow Your Body Clock. Imagine two identical twins standing side by side. They're genetically the same, born minutes apart, their DNA nearly indistinguishable. Yet despite these genetic similarities, one twin looks far younger, far healthier, and moves with a vigor the other has long forgotten. What creates this divergence? Researchers have found it comes down not only to genetics, but significantly to their daily habits. This fascinating discovery opens the door wide, inviting us to consider how simple lifestyle decisions influence our biological age, the internal clock that governs aging, health, and longevity. Of all the strategies scientists have studied to stretch the human lifespan, none have proven as consistent or convincing as caloric moderation, eating slightly less food. For nearly a century, countless studies, ranging from yeast and mice to primates, reveal that moderately restricting calories reliably slows the aging process. Animals on controlled calorie reductions consistently display fewer age-related diseases, brighter cognitive sharpness, and significantly longer lifespans. Yet, mentioning calorie moderation can raise alarm bells for many who associate it with hunger or deprivation. But here's the reassuring news in the human setting. Genuine caloric moderation doesn't mean going hungry. Rather, think of it as strategic timing and choices, not starvation. Instead of counting every bite of food, consider intermittent fasting, which is simply about adjusting when you eat, rather than severely limiting how much you eat. For example, simply narrowing your daily eating window slightly by skipping late-night snacks and early morning breakfasts can significantly lower inflammation markers, stabilize insulin levels, and stimulate beneficial metabolic processes. This structured timing encourages your body to use stored energy effectively, activating cleanup and maintenance pathways within cells. Just as you might tidy your home when guests aren't visiting, intermittent fasting grants your body quiet periods to repair, recalibrate, and refresh itself. Beyond timing meals, a second powerful tool is regular physical activity. Everyone understands exercise is essential for heart and muscle health, but the true magic happens deep inside each cell. Exercise is a signal, like an instruction message telling our cells, hey, it's time to upgrade the maintenance crew. With each workout, your muscles, bones, and organs kick into high gear, activating genetic pathways that enhance cellular repair and regeneration. In one particularly telling study, researchers followed over 650,000 individuals and discovered that even moderate physical activity, such as brisk walking, added anywhere from 3 to 7 additional biological years to a person's lifespan. Think about that instead of expensive anti-aging creams or mysterious pills. Simply walking swiftly most days significantly improved actual cellular aging. There's no mystery or magic. It's biology, responding positively to the signals it receives. Alongside physical movement, Your body performs another crucial maintenance operation nightly during sleep. Modern brain research has uncovered a remarkable cleaning mechanism known as the glymphatic system. While you sleep, activated brain cells shrink slightly, opening channels for cerebrospinal fluid to wash away harmful toxins, metabolic debris, and proteins that build up over daily use. Think of this as a dedicated night shift janitorial team clearing neurons to keep your brain operating sharply year after year. Missing this nightly reset consistently by regularly skimping on sleep not only accelerates cognitive aging, but contributes to overall bodily wear and tear. High-quality sleep helps slow that silent ticking clock of aging. Food quality, apart from quantity or timing, also profoundly impacts rates of aging. One dietary style standing out from decades of careful studies is the Mediterranean diet. It emphasizes colorful vegetables, legumes, whole grains, nuts, olive oil, lean proteins, fresh fish, and moderate wine consumption ingredients naturally rich in anti-inflammatory nutrients, phytonutrients, antioxidants, and healthy fats. Each meal gently nudges inflammation downward, strengthens cellular defense mechanisms, protects heart health, and supports cognitive sharpness. By adopting Mediterranean-style eating, your plate becomes a powerful ally in keeping your biological clock running slower. But food and exercise alone don't fully tell the story. Social connection and a sense of purpose also significantly influence aging. Scientists studying the so-called blue zones, certain places around the world such as Okinawa, Japan, Sardinia in Italy, or Nicoya Peninsula in Costa Rica, where residents consistently live significantly longer, have pinpointed something unique. Alongside diets rich in plants, fish, and healthy fats, communities in those regions possess strong intergenerational family ties, supportive communities, and meaningful daily routines. Elderly residents are actively engaged, socially connected, and fully integrated within their communities. Longevity isn't just about nutrients. Meaningful human connections genuinely enrich and lengthen life. Stress management intersects equally with healthier aging. Chronic stress, if left unchecked, substantially accelerates aging pathways. Elevated cortisol, the hormone our bodies produce under stress, triggers chronic inflammation, cellular breakdown, and accelerated tissue aging. However, countless studies confirm mindful practices such as meditation, yoga, deep breathing exercises, or simply quieting the mind dramatically reduce cortisol levels. By actively managing stress, your biological age quietly becomes younger without complicated medical interventions or expensive treatments. Let's take one remarkable example that illuminates the extraordinary benefits of lifestyle interventions. Researchers once examined a 40-year-old competitive marathon runner and found something striking. Due to consistent training, healthy diet, and disciplined recovery routines, this marathoner possessed arteries comparable to those of a healthy teenager. Rigorous aerobic exercise, clean eating, regular sleep, and careful stress management didn't just make him feel younger, it literally transformed his arteries, which are windows to the body's overall aging process. While promoting these lifestyle hacks, a cautionary note is important. In their eagerness to slow aging, many people turn to extensive dietary supplements, hoping megadoses of antioxidants and vitamins can reverse aging. Yet, large clinical trials repeatedly show that oversupplementation, particularly megadoses of antioxidants and isolated vitamins, offer no meaningful benefit, and in some cases, even increase health risks. Nature fine-tunes nutrients within whole foods, achieving a delicate balancing act not easily replicated or replaced by manufactured capsules. Sticking with unprocessed, whole food sources remains preferable for sustainable, healthier aging. Alongside this caution, certain non-negotiable basics remain critical foundations. While moderate alcohol consumption may have minor wellness benefits, Excessive drinking accelerates aging processes, damages tissues, and weakens organ health dramatically. Smoking is even clearer. It rapidly ages your skin, lungs, arteries, and cells, driving premature aging at every molecular level. Quitting smoking and moderating alcohol intake remain the foundational cornerstones upon which all other healthy lifestyles rest. To gauge the success of lifestyle interventions practically, easy-to-measure biomarkers can help track progress. Two particularly useful ones are resting heart rate and grip strength. A lower resting heart rate typically reflects strong cardiovascular health and effective recovery, reliably signaling an internal age that's progressing gently. Grip strength, meanwhile, strongly correlates with overall muscular health, resilience, and vitality. Tracking these easy, inexpensive biomarkers over months and years provides tangible reassurance that lifestyle changes are genuinely slowing biological aging. None of these approaches springs from novel high-tech laboratories, but each is remarkably powerful because of one essential factor: consistency. Aging gently and slowing your biological clock doesn't come from short bursts of dramatic effort, but rather simple lifestyle choices consistently applied over a lifetime. A lifetime of modest but steady improvements profoundly reshapes how rapidly your body's internal clock ticks forward. Lifestyle habits alone offer powerful aging interventions, but they are only part of the story. Now, biotechnology stands posed to accelerate and deepen what simple habits have begun. New approaches promise even more striking advances, including tailored genetic therapies and novel small molecules specifically designed to intervene deeper within cellular aging pathways. Biology's secrets are slowly coming into sharper focus, built upon lessons learned through simple lifestyle adjustments. In our next chapter, we'll investigate these deeper biological tools, exploring revolutionary new techniques promising not merely to slow, but perhaps even reverse our biological clocks entirely. Chapter 4: Gene Tweaks and Molecules That Turn Back Time. In 2020, in a modest laboratory cage, a mouse showed something quite extraordinary. At the start of this small experiment, it was visibly old, sluggish, with dull, patchy fur and struggling cognition. Yet, within weeks, this same mouse began to look youthful again. Its fur grew thick and shiny, its activity levels increased, and its mind sharpened. Scientists hadn't discovered magic. They had quietly achieved a remarkable biological feat. They'd essentially turned back the mouse's biological clock, reversing even, if just slightly, signs of age. But how could something so fundamental, so inevitable as aging, reverse itself? The secret to this unexpected result rested inside the genetic code. To understand this clearly, we'll start with a special toolkit discovered by scientist Shinya Yamanaka and his colleagues around 2006, work that later brought him a Nobel Prize. They identified just 4 specific molecules, known today as the Yamanaka factors: OCT4, SOX2, CALF4, and CMYC. When administered to mature cells in a lab, these factors reset cells effectively to a blank Sladia stem cell state. Cells that were previously skin, bone, or muscle became embryonic-like stem cells, capable of forming many different tissues. The mechanism behind them is now understood as epigenetic resetting. Epigenetics quite simply is the layer of instructions sitting above our DNA sequences. If DNA is a book full of written sentences, genes, the epigenome is the bookmarks or highlighter marks. It tells the cell which genes should be accentuated or hushed. Over decades, the epigenetic marks get scattered, misplaced, and noisy. When the Yamanaka factors entered the stage, it's like wiping all the messy bookmarks clean, returning the cells to an earlier, younger epigenetic state. But here's the catch. If you fully reset those cells to their embryonic state, they lose their mature identities. Worse, they can start dividing uncontrollably, leading straight into cancer territory exactly, where researchers never wish to go. However, scientists soon realized they didn't have to go that far. Instead, they discovered partial reprogramming. Think of partial reprogramming as gently nudging the cell's epigenetic clock backwards just enough to erase age-related marks without changing the cell's identity drastically. Partial reprogramming improves function in tissues and organs, offering rejuvenation without tipping into uncontrolled growth. Yet aging isn't only about epigenetic bookmarks. It's also about accumulating small mistakes in the DNA like typos, in a sentence slowly building up until meaning becomes distorted. Consider progeria, a heartbreaking premature aging condition caused mainly by a single DNA letter error in one crucial gene. Recently, scientists used an advanced gene editing technique known as CRISPR base editing to fix this very single letter error in lab-grown human cells. Unlike traditional gene editing tools that cleave and repair DNA strands, CRISPR base editors precisely rewrite individual genetic letters without cutting both strands. This gentle yet precise approach greatly reduces unintended risks, showing promise not only for progeria, but potentially correcting other DNA spelling errors involved in aging. Another facet of aging is zombie cells, technically called senescent cells. These cells stop dividing a useful safety mechanism in youth but in old age. They accumulate dangerously, spewing harmful signals into surrounding tissue. These cellular zombies cause inflammation and tissue breakdown, accelerating aging. Researchers have devised senolytic drugs to specifically detect and remove these senescent cells. One promising combination is dasatinib, previously approved cancer drug, and quercetin, which appears naturally in apples and berries. In elderly mice, the combination consistently improved heart function, lifespan, and maintained vigor. Human trials today cautiously explore these findings with early signs of success. Then comes NAD, nicotinamide adenine dinucleotide molecule, found in abundance in younger cells, responsible for fueling cellular repairs, and mitochondriate cells' energy-producing batteries. NAD declines steeply as we age, leaving cells less capable of energy production and self-repair. Scientists found NAD boosters, like the molecule NMN, nicotinamide mononucleotide, could effectively replenish NAD supplies. Animal experiments report enhanced metabolisms, clearer cognitive ability, and improved muscle and bone strength with NMN supplementation, fueling considerable hope and a somewhat contentious market for human supplements. Scientists have also long known calorie restriction, eating fewer calories without malnutrition, extends healthy lifespan across nearly every organism studied. But strict dietary limits are notoriously hard to maintain. Enter rapamycin, a molecule initially discovered in soil bacteria from Rapa Nui, known to many as Easter Island. Rapamycin targets a key growth pathway called mTOR, mammalian target of rapamycin. Much like calorie restriction, rapamycin puts the cellular growth programs into maintenance mode, encouraging cells to repair and recycle rather than grow rapidly and age. The effect: mice fed low-dose rapamycin lived significantly longer lives with improved immunity, heart function, and cognitive resilience. Even elderly family pets, in early trials with older dogs given low rapamycin doses, showed remarkably increased vitality and extended lifespans. Perhaps the most striking and controversial aging interventions involve blood itself. Earlier experiments called parabiosurgically, joining an old mouse's circulatory system to that of a young mouse, showed improved muscles, heart function, and learning ability in aged mice. But giving older humans younger blood directly raises significant ethical and medical questions. Scientists now isolate beneficial molecules or plasma fractions from young blood, offering a safer, targeted path forward. Early studies are intriguing, yet require larger, rigorous testing. Restoring youth to tissues worn by age often focuses on stem cells. Aging depletes stem cell reserves, particularly in cartilage and the immune system. Stem cell infusions aim to replenish these crucial cells. For joint cartilage, replenishing stem cells can reduce pain, repair worn-out tissues, and even restore more youthful movement. Immune system rejuvenation, with replenished young stem cells, can likewise restore defenses against infections and cancer, opening up potentially healthier elderly years. How do scientists objectively test if these methods succeed? One reliable yardstick is our biological age, determined by DNA methylation clocks, measures based on exact patterns of epigenetic bookmarks. After successful anti-aging therapies in animal models, these clocks consistently turn backwards, reverting tissue and biological age to youthful markers. Researchers now closely measure these methylation clocks in human tests, cautiously optimistic. Yet excitement must be tempered with realism. Each promising intervention faces obstacles. Regulatory authorities like the FDA scrutinize new therapies carefully, especially those affecting foundational sector processes like aging. Moreover, the cost to consumers currently limits who might benefit from these modern medical marvels. Despite online marketing hype, we must carefully acknowledge what we don't yet know clearly. Long-term, unintended complications, effectiveness across diverse human populations, and life course impacts from a lifetime of treatment. Weighing risks versus benefits is crucial. Epigenetic resetting and partial reprogramming could inadvertently open risk pathways toward cancer, despite careful controls. Gene editing methods correcting DNA might carry unforeseen side effects due to off-target effects or unexpected genetic interactions. Even NAD boosters, despite their apparent safety, lack lengthy and conclusive data in human trials. Rapamycin, thoroughly studied in mice, remains only partially explored at low doses in aging humans. Similarly, senolytics and plasma fractions have potentially remarkable value, yet uncertainty lingers about long-term safety. Only rigorous, randomized, controlled human trials can definitively determine safety and effectiveness. These carefully designed trials demand patience and rigorous scientific objectivity, free from promising but unproven hype that saturates the wellness marketplace. Before revolutionary treatments become mainstream, science owes everyone transparent evidence and clearly communicated risk information. Finally, even as these tools emerge from laboratories into clinical possibilities, they inevitably pose challenging social and ethical questions. Should anti-aging strategies become publicly accessible medicine or remain available only to the wealthy? If we extend lifespan dramatically, how does society adapt to the resulting population dynamics? And ethically, if aging is altered, what implications arise about natural lifespans and healthcare fairness between generations? Undeniably, exciting biological tools to fight aging emerge today, promising genuine hope. But science must never advance recklessly without reflective thought, transparent caution, and thoughtful ethical awareness of the profound human impact at stake. Chapter 5. Risks, Ethics, and the Road Ahead. Imagine a future where job interviews are attended not by fresh college graduates in their 20s, but by vibrant individuals celebrating their 120th birthdays. A world where retirement doesn't mark the end of a career, but rather the starting line for a new chapter of professional life. It sounds astonishing, perhaps even inspiring, but raises important, challenging questions we need to address today. One question quickly comes into view. Who has access to these groundbreaking treatments first? Would we see the billionaire entrepreneur living comfortably into their second century, while the average teacher, construction worker, or nurse must wait as their health continues to decline? If history is any indication, revolutionary medical advances often reach the wealthy first, before becoming accessible to all. You only have to look back to early transplantation surgery and advanced cancer treatments to understand that economics, ethics often dance a very complicated dance. Now let's consider a society where life expectancy rapidly extends for virtually everyone. At first glance, this scenario might seem idyllic, a society where we witness our great-great-grandchildren entering adulthood. But think practically. Our existing economic and social systems aren't built to cope with enormous jumps in longevity. Pension plans, social security, and health systems could come under tremendous strain. Pension systems rely on financial models built around predictable lifespans. Extend those years substantially, and suddenly, countries everywhere face profound financial challenges, perhaps even crises. Hospitals and medical centers, already stretched thin in many regions, would need to adapt quickly and at great cost to accommodate soaring demands for care over many more decades. There's also that elusive yet vital question of population. Some worry that if people live significantly longer, we might see an unsustainable increase in population, straining resources further. But this concern might soften when we look closely at current global trends. In countries where average lifespan increases, birth rates often decline. Japan, Italy, and several northern European nations are prime examples of what demographers call demographic transitions. When people feel confident their children survive comfortably into adulthood, when healthcare is robust and lifespans are longer, births tend to slow significantly. Perhaps, instead of crowded cities and resource depletion, a longer living population could lead to a balanced scenario-moderated birth rates combined with longer, healthier lives. Yet even with balanced demographics, consider how the deep fabric of families and communities might shift when 4 generations live simultaneously. Imagine visiting grandparents who themselves care for their own elder parents. It's a reality many of us might experience, and one that introduces significant cultural shifts in our relationships, family dynamics, and support systems. How would roles and responsibilities transform in this scenario? Would seniors find new identities and purpose in long careers? Or would younger generations face new struggles as family members live actively into extreme old ages? Beyond social impacts, we must grapple with significant biological realities and safety risks. If our interventions in aging biology become too aggressive, we may invite unintended side effects, some potentially severe, such as increased risks for cancer. Cellular reprogramming technology zone of the promising but experimental approaches to extend lifespans work by reversing cells back toward youthful states, but push too far and cells could lose their stability, increasing the very dangers we're trying to mitigate. Researchers continue to be cautious because history teaches us definitive warnings. For instance, early hormone replacement therapies in the 20th century were initially considered miracle treatments, celebrated widely as fountains of youth. Yet frustratingly, years later, researchers discovered these therapies significantly increased risk of heart attacks and stroke. It was a painful lesson. Interventions, no matter how promising they seem, must undergo rigorous testing and scrutiny before broad adoption. This brings us to another critical concern: the slippery and dangerous slope of unregulated clinics and unscrupulous sellers promising miracle anti-aging cures. Each day, the internet buzzes with marketing geared toward desperate clients craving longevity solutions, miracle cocktails, unregulated stem cell infusions, and premium-priced rejuvenation products. Historically, whenever groundbreaking medical advances make headlines, such dubious actors swiftly emerge, advertising wonder therapies well beyond reliable evidence. Inevitably, without proper medical oversight, these unproven interventions not only drain financial resources, but pose significant threats to patient health and safety. Fortunately, robust regulatory frameworks exist to safeguard us. Regulatory agencies like the Food and Drug Administration, FDA, in the United States, and the European Medicines Agency, EMA, in Europe, have adapted rigorous frameworks carefully tailored for aging therapies. They mandate exhaustive clinical trials, emphasizing patient safety, clearly demonstrated medical benefits, and thorough evidence-based standards, ensuring any anti-aging treatments that reach consumers stand on solid scientific ground. Though some criticize stringent agency oversight as overly cautious and slow, such safeguards function as necessary gatekeepers, preventing rash enthusiasm from overshadowing well-measured caution. But perhaps the most profound debate in the science of aging revolves around a deeper philosophical question. Should aging be viewed as a treatable disease, something we constantly strive to cure? Or should we accept aging as a natural process that shapes and possibly enriches our human existence? Many researchers passionately advocate classifying aging as a genuine disease, arguing this perspective opens avenues for intensive research and creative solutions. On the other side, some ethicists and scientists caution against seeing aging strictly as a medicalized problem, emphasizing instead a deeper appreciation of the aging process itself, embracing its dignity, wisdom, and meaning, rather than seeking purely to overcome it. This philosophical debate brings us to an important concept: health span. While much public discussion focuses on lifespan, actual number of years lived in, increasing number of experts emphasize health span. Health span refers to the years of healthy, active living a person experiences. The real goal of modern aging research isn't just about adding empty years spent in sickness or disability, but adding vibrant, healthy years filled with experiences, joy, and independence. Quality of life improvements matter equally, if not more, than extending lifespan itself. This nuanced and balanced approach resonates with many people globally. Public opinion surveys frequently find cautious optimism surrounding longevity research. Many express excitement at the promise of healthy aging breakthroughs, yet voice concerns about potential side effects, equity of access, and unforeseen social impacts. Overwhelmingly, most indicate openness toward treatments that improve quality of life, maintaining independence and energy into advanced ages, but remain wary of interventions promising immortality or extreme lifespan extensions. So how should you respond to the swiftly evolving landscape of anti-aging treatments and therapies? Here are some practical, scientifically grounded guidelines. First, maintain a healthy skepticism toward bold claims of miraculous rejuvenation, especially when presented outside reputable clinical institutions and medical journals. Second, base your health decisions firmly on rigorous scientific evidence and clinical trials, not celebrity endorsements or marketing campaigns. Third, always prioritize medical consultation with trusted professionals and healthcare providers before embracing new therapies or experimental treatments. Looking forward, it's thrilling yet reassuring to picture a future shaped deeply by precision medicine, personalized strategies crafted specifically for individuals according to their unique biology, genetic markers, and lifestyle preferences. Instead of generic, standardized treatments, imagine doctors offering highly tailored longevity and wellness plan strategies precisely matched to each individual's health status, genetic makeup, and personal goals. Such a personalized approach, harnessing the power of rigorous research combined carefully with individual differences, may provide a hopeful way forward. Ultimately, while the path forward comes bundled with genuine ethical questions, social challenges, and medical uncertainties, there remains genuine optimism grounded in responsibility. Throughout history, humanity has shown extraordinary capacity to use scientific breakthroughs wisely to improve human conditions while maintaining ethical and equitable principles, despite inevitable missteps. Our journey into extending healthy lives is no different. By embracing this blend of careful optimism, scientific rigor, and thoughtful responsibility, our future becomes a realm not of fantasy, but of grounded possibility. place where longer, richer, healthier lives become genuinely achievable reality for all of us.